Okay, so the final part of macromolecules is to look at the nucleic acids. And nucleic acids, nuclear, refers to the nucleus inside the cell, generally in the center of the cell. Nucleic acids are what you find inside there, and there are two types. There is DNA and RNA. So our DNA and our RNA, I didn't write out that one there, so I'll do it now. So these are the two found in nature. Now, again, like carbohydrates and like proteins, nucleic acids have monomers, meaning the individual unit. And the monomer here is the nucleic acid itself. So what's it made up of? It is made up of three parts. It's made up of a phosphate group, and attached to the phosphate group through a phosphodiester linkage, which I've written here. That's the phosphodiester linkage there, is a sugar group, and these make up the backbone. So you can see both in the RNA molecule here and the DNA molecule I've drawn here. That's the side of the ladder. If you're going to draw a ladder, put the rungs in, well, we're talking about that part there. So this is the backbone of both of the molecules. So you have sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the same in both of them. But there is one significant difference in that backbone, and that's the sugar. And that's the reason why the names are different. Both of the sugars are ribose sugars. Now, ribose sugar equals 5 carbons in it. The only ribose sugars found in nature, or the only 5 carbons, sugars I should say, found in nature are ribose sugars. And there's two versions of it. There's the ribose sugar itself and the deoxyribose sugar. So in this sugar molecule it has one less oxygen. So it's a deoxy, one less oxygen in the ribose sugar. So they're structurally very slightly different. So the deoxyribose sugar is the backbone of the DNA, and the ribose sugar makes up the backbone of the RNA, hence the name deoxyribose nucleic acid and ribose nucleic acid. So there's our backbone. Sticking out, the third part is the base. And there are 4 bases in both. So for DNA, the short bases, because this is a 1-ring structure, and these are 2-ring structures. These are not accurate in terms of their structure. I just want to give you the impression of what I'm talking about. So 2-ring structures, 1-ring structure. That makes more sense when we actually talk about it in a few weeks' time. So in DNA you've got cytosine, adenine, thymine and guanine as your 4 bases. In RNA, you have cytosine, adenine, and guanine, the same as DNA, and then a difference here, uracil replaces thymine. So differences between DNA and RNA are quite clear. There's a difference in the sugar. This is deoxyribose, this is ribose, and I've written that here, DNA, nucleotide, deoxyribose sugar, RNA, ribose sugar. DNA is like a ladder. It is double-stranded, so you've got a side coming down here and a side coming across here. So it's double-stranded. RNA is just single-stranded, meaning that the bases, and these are the things that carry the information on an RNA, is exposed and it's protected in the DNA. And the final difference is the base itself. Thymine is replaced by uracil in RNA, but they're both nucleic acids. They're joined in the backbone. The monomers are joined together by a phosphodiester linkage, and we will look in much more detail in a few weeks' time about how these are then transcribed and translated into proteins. So there's a nice short one. Thank you for listening.